Hi everyone, my name is Marielena Grijalva. I had the excellent opportunity of interning at Dr. O'Neill's lab this summer at the University of Pennsylvania. My presentation is on developing a Singulator protocol for nuclei isolation in uterine tissue. Just as a disclaimer, my presentation does include a picture of a uterus before dissection. In partnership with HuBMAP, our lab is generating a single-cell molecular map of the human reproductive system. We are obtaining whole human reproductive systems from donors and systematically dissecting them to create spatial samples for single-cell molecular assays. The O'Neill lab focuses on endometrial regeneration and factors impacting the stem cell niche in the endometrium. There are 3 different organs in the female reproductive system, each with a different tissue density. You can associate tissue in different ways, which can affect the tissue cells. We are processing tissue pieces that have very different characteristics. The ovaries have follicles that are filled with fluid and eggs. Then you have the fallopian tubes that have very little muscle. The uterus is conducted of the myometrium, endometrium, cervix, and the uterine body. The cervix has fibrous tissue, which can result in a tissue tough-like consistency. Any molecular assay that we do is going to require cell dissociation, and there are different ways to dissociate tissue. There's mechanical and enzymatic, and sometimes both. This is a uterus we received at the lab. We dissected the uterus into small pieces and then snap froze the tissue pieces. Both the manual and Singulator protocols use snap frozen tissue to obtain nuclei suspensions. The Singulator offers reproducible, rapid, and hands-off tissue dissociation into a single cell or nuclei suspension. The Singulator protocols are preset, and there are protocols for cell and nuclei dissociation. There are customizable parameters, including mincing, enzyme incubation time, temperature, mixing, and mechanical disruption profile. Prior to my arrival, many manual protocol runs of the uterus and cervix were not successful. In an effort to get better results, we started to use the Singulator for uterine and cervical tissue. The goal of the protocol is to result in a nuclei suspension with high nuclei yield, low clumping, low blebbing, and low debris. Blebbing is when the nuclear membrane changes shape and is no longer oval or round. Clumping is formed when the nuclei membrane fuses together and makes a large clump of different nuclei. The Singulator protocol is more time efficient and also has lower sample loss. There is more human error involved in the manual protocol since we have more control over each step of the experiment. The Singulator allows for better temperature control throughout the entire experiment. Running one trial with the manual protocol is more cost efficient, as with the Singulator protocol, running multiple trials over time becomes more cost efficient. In the picture on the left is all the equipment you would need to run a manual nuclei isolation protocol. In the picture on the right is the equipment needed to run a Singulator nuclei isolation protocol. All human subjects gave informed consent to participate in the study. Participants for the optimization samples were recruited from the Hospital of the University of Pennsylvania, HUP. Most of the participants were on testosterone treatment undergoing gender affirmation surgery, but were otherwise healthy. Samples for optimization were collected from 2020 to 2021. Samples were taken fresh and then snap frozen on dry ice. The samples were then stored at -80 degrees Celsius until they were ready for use. The following section covers the trials we did to optimize nuclei isolation in cervical tissue. We ran 6 cervical trials on the Singulator. I adjusted the parameters and protocols of the runs to clear up blebbing, debris, and clumping. We ran trials with the extended nuclear protocol as well as the nuclei low volume protocol. The extended protocol allows for a longer incubation period. We ultimately decided to stick with the extended protocol since it gave a better nuclei yield. We also switched from a 40 micron strainer to a 20 micron strainer to lower the debris, and we changed the incubation time to 5 minutes. The picture on the right shows a piece of debris in one of our cervical nuclei suspensions. On the left, you can see that I have labeled a nuclei with a solid membrane, and I've also labeled a nuclei with blebbing. Here you can see the final parameters of the Singulator protocol that I have developed for cervical tissue. The portions that are highlighted are what I adjusted for the default nuclear protocol. Also, out of all the images captured from the cervical nuclear suspensions, this one is my favorite. This following section covers the trials we did to optimize nuclear isolation in uterine tissue. We ran 9 uterine trials on the Singulator. We ran trials with the extended nuclear protocol as well as the nuclear low volume. We chose to stay with the extended protocol due to its higher nuclear yield. We also adjusted the straining from using a 40 micron strainer to a 20 micron strainer. We changed the incubation time from 7 minutes to 4 minutes, but ultimately we landed on a 5-minute incubation time. In the picture on the left, you can see a cell with a solid membrane, as well as a membrane with flapping. The picture on the right shows a piece of debris from a uterine nuclear suspension. Here you can see the final parameters of the Singulator protocol that we developed for uterine tissue. The highlighted portions are what we altered. The differences between the final parameters of the uterine and cervical tissues is the mixing speed. Cervical tissue has a tougher consistency and requires the fastest mixing speed and the fastest disruption speed. Thank you to the NIH for this amazing learning opportunity. I am forever grateful for the chance to spend my summer doing groundbreaking research at one of the top schools in the country. I'd like to thank Dr. Kate O'Neill and Addie Johnston for welcoming me into the lab, as well as mentoring me with patience and kindness. I would also like to thank Jean Rosario and Hugh Nguyen from Dr. Jungyeon Kim's lab for going above and beyond as mentors and as amazing friends. We thank all of these anonymous patients for participating in this research. Lastly, I'd like to thank my mother and father for immigrating to America so I could have a better future. I could not have come this far without their love and support of my educational goals. Thank you, everyone.